Hello everyone, in this video we are going to discuss about the RNA splicing. So before discussing about the RNA splicing, first of all we should know how this RNA is formed. What happens, we have the genomic DNA and this DNA is present inside the nucleus, and by the process of transcription what we get is we get the pre-mRNA. It is not the mature mRNA, it is the pre-mRNA. That means we are having both the exons as well as in the introns. So what we have to know, what is the intron and what is the exon. So let us first be clear with this term. We have a term known as exon, and this exon is the coding region of RNA. Okay. Next comes the term intron. This intron is the non-coding part of RNA. So what happens, here we have a DNA, this is a DNA, and from this DNA we get a RNA or mRNA. From this mRNA we get amino acids. When this amino acid will join together, they will form a protein. Okay, and the formation of DNA to RNA is by the process of transcription, and the formation of proteins is by translation. Okay, so what happens when the transcription is taking place, both the exons as well as the introns are being transcribed. There are various type of introns as well as exons. Okay, so here what we got, here we got first exon, next intron, exon, then intron. So what happens in the splicing mechanism, we have to remove the non-coding region. That means we have to remove the introns. Only exons should be present in the mRNA after the splicing, so that it will give the coding region, or the all the things which are present in a RNA will help in the formation of a protein. So what we have to know is what are the processes of splicing. We are here discussing about the RNA splicing. There are variety of RNA splicing. So one by one we will discuss, and in this video we are going to discuss about the normal splicing. So let us first know the types of splicing. The splicing are of three types. So what is splicing? The removal of introns. The removal of introns are known as splicing. Okay, are known as splicing. So there are three types of splicing. First is the normal splicing. What happens in a normal splicing? There are various types of spliceosomes involved during this process, like the U1 snRNP, U2, U4, U5 and U6. These are the snRNPs. Okay, or we can also write as SNRPs. Okay, the next type of splicing is trans-splicing. Trans-splicing. And the third type of splicing is alternative splicing. In this video we will discuss about the normal splicing. In the other video we will discuss about the trans-splicing as well as the alternative splicing. So what we have to know first is what is the difference, that it is a exon or it is a intron. Say here from this diagram you can be very clear that the introns are present in much amount when compared with the exon. Okay, so we have to remove this introns, and this intron is the non-coding region. What happens, there is a enzyme which is known as dihydrofolate reductase. There is a enzyme which is known as dihydrofolate reductase. This is a enzyme, and this enzyme is more than 31 kb long. But what happens? The intron part is more in this dihydrofolate reductase enzyme. The only exon is 2 kb long. That means we are having 31 kb long enzyme which is known as dihydrofolate reductase, but in it there is only 2 kb of the expressing part, that means the exon. So you can now see how the intron is taking the space. So we have to remove the introns, only we have to keep the exon. That means only the coding part should be remained. Okay. So what is the difference that it is a exon or a intron? As we know that in the RNA we are having a 5' region as well as a 3' region. The area near the 5' end is known as 5' splice site, and the region near the 3' end it is known as 3' splice site. What happens here in the intron? We are having a GU region near the 5' splice site. What happened near the 5' splice site? We have GU rich region, and near the 3' splice site we have AG region. So where we have GU and AG repeat as a conserved sequence, we have near the intron. See here, it is this part where we have GU and we are having here AG. These are the conserved region and these are present near the intron. And in the intron we have another site which is known as branch point. Which is a branch point? Adenine is a branch point. So if there is A, see here we are also having A, but this is not the branch point. So how we can recognize that this A is only the branch point? We can recognize when adjacent to it is the pyrimidine. If adjacent to the branch point or adenine, we have pyrimidine, then we call it as branch point. Okay, or we can also write as, if adjacent to the A, we should not write here branch point. If adjacent to adenine, if we have pyrimidine, that is known as branch point. So we are clear with these three terms. So we are having another term here, that is the 5' splice site is the donor site and the 3' splice site is the acceptor site. Okay. Now let us see how this process of splicing is taking place. See here, we are having the branch point, that is A. Here we are having the branch point, that is A. What is A? It is adenine. What happens in case of RNA? The 2' end and the 3' end are present. That means OH is present in the 2' carbon. Say, let us draw the structure of this RNA. Here it is the pentose sugar, and here nitrogen base adenine is being attached, and near the 2' and the 3' OH group is present. So this OH group of the 2' end will help in a reaction which is known as nucleophilic attack. This 2' OH of adenine will do a nucleophilic attack. Where it will attack? It will attack in this region. In the 5' splice site or the 5' end, it will form a nucleophilic attack by which there will be a cleavage. Here there will be a cleavage. When there will be a cleavage, here what happens? This introns will be removed from this exon. So, after the removal of this introns from the exon, it will form a lariat-like structure. This structure is known as lariat-like structure. Okay. So, if there is a cleavage, we are having here exon, which is the coding part. Okay. So, this is the 5' splice site and opposite to it will be the 3' OH group. Okay. Now, what happens in the 3' OH group? Now, this 3' OH group will help in making next nucleophilic attack. Where it will do its nucleophilic attack? It will do near the 3' splice site. So, when it will do the nucleophilic attack, here there will be a cleavage, so that this lariat-like structure or the introns will be removed completely in a lariat-like structure. You can see here the total lariat-like structure, which is the intron, is being removed, and here we have two exons ligated. This axon and this axon is being ligated because here we are having 3' OH group and here we are having 5' phosphate group. This 3' OH group will be ligated with this 5' phosphate group. So, what are the points here? Here we are having 2' OH group, and this 2' OH group act as nucleophilic attack. That means nucleophilic reaction is taking place, and for this process to be done, there are two transesterification reaction. Okay, so what is two transesterification reaction? The nucleophilic attack which is performed, that is known as transesterification reaction. The first transesterification reaction is when the 2' OH group of adenine or the branch point is making a nucleophilic attack with the 5' splice site, and the next transesterification reaction is when this 3' OH group is making a nucleophilic attack with this 3' splice site. So that we will get here a lariat-like intron and the separated exon we can get. So this is all about the overall what is splicing, what is the reaction taking place when the splicing mechanism is taking place. So for reading this or for learning this splicing mechanism, there is involvement of various type of proteins. Okay, let us look what are the proteins. See here, these proteins which are involved in the splicing mechanism, they are known as the spliceosomes. Okay, so what are the spliceosomes? They are also RNAs. Okay, so there are involvement of 5 RNAs. What are they? They are the U1, U2, U4, U5 and U6 RNA, or we can also write it as snRNAs, small nucleolar RNAs. snRNAs means small nucleolar RNAs. So, when this U1, U2, U4, U5 and U6 will come and bind, as we know that this U1 is also a RNA, U2, U4, U5, U6 are all RNAs. So, when they will come and bind to the mRNA or the pre-mRNA, how will they bind? They will bind with the complementary region. These U1, U6, U2 all are the RNAs. So, they will have some of the sequence which will be similar to the mRNA. in that place only they will come and bind. Okay, so the U1 will come and bind near the 5 prime splice side, and the U6 will also come and bind near the 5 prime splice side, and the U2 will come and bind near the branch point. You can see here there is a branch point where the U2 is coming and binding, and the BBP will also come and bind near the branch point. So what is the BBP? It is the branch point binding protein. BBP means branch point binding protein. Okay. So this U6 and U2 are also the RNAs. Okay. So let us see the mechanism how this is done. The U1 snRNPs will come and bind to the 5 prime splice site, and the BBP or the branch point binding protein will come and bind near the branch point, that is near the adenine. Along with it there will be U2F65 and 35 which will be adjacent to the BBP, and this complex is known as E complex. Okay. It is also known as commitment complex. So the U2AF which is present, this U2AF which is present, it is having two subunit, that is the 65 subunit and the 35 subunit. What happens? We have already discussed that adjacent to this adenine, that is pyrimidine. Okay. So this 65 which is present, the subunit of U2AF, that is 65 subunit which is present, it binds with the pyrimidine track. Okay. And the 35 subunit, it bind near the 3 prime splice site. Okay. So what happens next? This is known as the E complex or the commitment complex. In the next step, what happens? There is a recruitment of U2 snRNPs. When the U2 snRNP will come, the BBP or branch point binding protein will be removed. And this stage is known as pre-spliceosome complex. Okay. When this U2 snRNP will come, this BBP is dissociated or it will be removed. So it will take place of BBP. U2 snRNP will take the place of BBP. But see here there is a difference. When the BBP was present, this A was in a straight line, but here there is a formation of loop. Okay. In the next step, what happens? There are 3 snRNPs coming. What are they? They are the U6, U4, U5. When these 3 will come, the U2AF which was present, it will be removed. So U6, U4, U5 came here. Here this is U4, this is U5 and this is the U6, and near to it is U1. With the recruitment of U6, U4 and U5, the 5 prime splice site will come near the branch point. It will help in bringing the 5 prime splice site near the branch point. You can see here it was very long, but with the recruitment of this protein, here the 5 prime splice site is near the branch point. Okay, and this complex is known as B complex. It is known as B complex or spliceosome complex, because all the spliceosomes are present in this region. Okay. So what happened next? The U1 which was present near the 5 prime splice site now dissociates or it is removed. With the removal of the U1, what happens? The U6 now pairs with this 5 prime splice site. Okay. In the next step, the U4 which was present, it will be dissociated or it will be removed from this complex. So, with the removal of U4, the U6 will bind with the U2 SNRNPs. When this U6 will bind here with the U2, there will be a formation of lariat-like structure. The cleavage will take place over here. Okay, what happens? As we have discussed that here, first of all, we are having a branch point of which the 2' OH group was making a nucleophilic attack to the 5' splice site. Okay. See here, we are having here the 5' splice site was very far away. That is the reason we have to bring the 5' splice site near the branch point, so that from here the 2' OH will help in the cleavage, or there will be nucleophilic attack, because to the very long distance the nucleophilic attack cannot be done. So, this 5' splice site will come near the A site, that is branch point, near the branch point, due to which there will be transesterification reaction or there will be nucleophilic attack. This A which is present, the 2' OH group of this A will help in the formation of this cleavage or nucleophilic attack will be done over here. When the U4 is dissociated, this nucleophilic attack is done. When the U6 and U2 will combine, see here in this region the nucleophilic attack is done. So, when this nucleophilic attack will be formed? When the U4 is being removed and the U6 and U2 comes in close contact, during that time there will be a nucleophilic attack. This A will make a nucleophilic attack, the 2' OH group of this A will make a nucleophilic attack to the 5' splice site, so that there will be a formation of lariat-like structure. Okay. So, what happens here? We got, if this is a 5' splice site, we here got 3' OH group in this region. This 3' OH group will now make next transesterification or nucleophilic attack in the 3' splice site. So, here there will be next nucleophilic attack and here there will be a cleavage. If there is a cleavage, this lariat-like structure will be now removed. See here, we have got this intron which is in the form of a lariat-like structure. Including the introns, there are various type of SNRNPs, that is the U5 SNRNP, U6 SNRNP and U5 SNRNP, and we have got the exon which is the coding part. So, what happens? Here we have lariat-like structure intron, this is the intron with lariat-like structure, as well as we have SNRNP. So, this SNRNP should be removed, so that in the next splicing mechanism it can take part. So, the dissociation of this SNRNP is done by PRP22, or it is known as dead box helicase. Okay. So, this type of proteins or the SNRNPs is used when the mRNA is very long. But if we have a short mRNA, it is done by cell splicing. The cell splicing are also of two types. They are the group 1 cell splicing and the next one is the group 2 cell splicing. When we see about this group 2 cell splicing, here we are having group 2 cell splicing. It is similar to the pre-mRNA spliceosome mechanism. Here what is the only difference? The only difference is that here we have some of the spliceosomes, that means here proteins are being involved, but in group 2 cell splicing proteins are not involved, they are making the cell splicing. Here near the branch point, here we have a branch point, the 2' OH group of this branch point make a nucleophilic attack in the 5' splice site, and here we will get 3' OH group. Here we are getting here 3' OH group. This 3' OH group will make a nucleophilic attack with this 3' splice site, and we can get a lariat like structure, and the exon will be present over here and they will be ligated. This exon and this exon will be ligated. And when we discuss about the group 1 self splicing, what happens? There is a G region over here. In the group 2 self splicing and spliceosome mechanism, we were having A. Adenine was present and 2' OH group of this adenine was taking part in the nucleophilic attack. But in case of group 1 self splicing, we have guanine. In the branch point, we have guanine, and the 3' OH of this guanine is taking part in the nucleophilic attack in the 5' splice site. So, when there will be a nucleophilic attack in the 5' splice site, we will get 3' OH group over here, and there will be cleavage of this intron from this 5' splice site. Here, this 3' OH group will help in making another nucleophilic attack in the 3' splice site, and here there will be removal of the intron. So, this is not in a lariat like structure. The group 1 self splicing will not form the introns in the lariat like structure. It will be in the form of a nearly linear shape, and the exon will be ligated together. Okay, this is the main difference between the pre-mRNA spliceosome, group 2 self splicing mechanism and the group 1 self splicing mechanism. Thank you for watching this video till the end, and if you like this video, please do like, share and subscribe. Thank you.